Hello, now we are going to start with chemistry and structure part 2. And with this series of presentations, we will be talking about arabinose islands. Again, as you know, we refer them sometimes non-starch polysaccharides or pentosanes. And we will talk about beta-glucan and starch. Starch is extremely important for cereal chemists and for us, and then total starch determination, and then also wheat lipids. If we have an understanding of basics of each compound in wheat kernel, again, as crop quality specialist or technologist, cereal chemist, cereal scientist, we can have a better understanding of how to modify our system, what really the data means. Again, we know from our previous lectures that wheat kernel is very complex, and then when we extract endosperm, we know each part of the endosperm, each meal fraction will have different chemical composition. So now we will go more in depth what are the wheat carbohydrates and what are their chemistry, their functionality, and why they are so important. Again, if you look at the wheat grain, and I know we spent so much time discussing proteins, spring wheat protein is the functional protein, protein quality is extremely important. But the most abundant carbohydrate, the most amount of compound in the wheat grain is carbohydrate actually. Why? Because we have 60 to 65% starch in endosperm, and sometimes it's a little higher for soft wheat classes. And then we have 8 to 15% proteins, and lipids are ranging from 2 to 4%. And then we have minerals and vitamins 1 to 3%. When we say carbohydrates in wheat, these are the carbohydrates we found in wheat. Starch is the most abundant, and arabinose islands. From now on, I'm going to refer them as arabinose islands. We use the terminology interchangeably, non-starch polysaccharides of wheat or pentosanes, but again, I will be using the word of arabinose islands. After starch, arabinose islands are the most abundant for wheat, and the next one is beta-glucan. We can have smaller sugars in wheat, and cellulose is very low amount, and most of the time it's mostly located in bran. And then we can also have another class of polysaccharide is glucofructans. Again, we will spend some time on what they are. So we talked about wheat bran and endosperm and what we have. So starch is the major source of energy stored in the wheat grain, and then we save that the wheat is saving that energy until the germination starts. We will link this to pre-harvest sprouting later in the lectures. And then the carbohydrates are produced through photosynthesis. So we know the plants are taking the carbon dioxide and all nutrition from soil, and with the daylight they are converting everything else to carbohydrates and different compounds, but the carbohydrates are produced through the photosynthesis. The photosynthesis is responsible for conversion of carbon dioxide into the organic polymers annually. So why again carbohydrates are so important? This is something I really like because it's the chain reaction. Again, if we know the details of carbohydrates, because if they are small or big, if they found in the nature in this fashion or that fashion, or how their structure is impacting how they are going to behave in the food system. For example, viscosity. You know, we all run amylographs or rapid viscoanalyzers or farinographs, and sometimes you see changes, less viscose, more viscose. Everything is impacted by all these profiles are controlled by the details in the chemistry. And then once we know that, we know it's going to change rheological behavior, but then at the end it's going to impact the function. That function will be responsible of the texture of the final product. Is it crispy? It's soft? What's the shelf life? Again, linking properties to application is very important. Again, this is why we go a little deeper into this part, so we can understand why chemistry is so important, how that's linked to the final quality. So for arabinose islands are the major non-starch polysaccharides again in wheat. As you can see in this figure, wheat has very high percentage of non-arabinose island, followed by barley and rye, and rye actually has very high amounts of arabinose islands and other cereals. So for each cereal, there is major non-starch polysaccharide. For example, for wheat and rye, arabinose island is the major non-starch polysaccharide, but for barley and oat is beta-glucan. So we have lesser amounts of beta-glucan in wheat compared to oat and barley. Okay, this is the final structure of arabinose island. So arabinose island is a beta 1,4 linked, we call that pentose. And why? Because it's a 5 carbon sugar. If you think Latin is pentose, penta means 5, so pentose sugar is 5 carbon sugar. That's why arabinose islands are referred as pentosanes, because of 5 carbon. If you look at here, these are how we are numbering the carbons. For example, 1, 2, 3, 4, and 5. The sixth member in the ring is oxygen, so we have 5 carbon. So arabinose island, the backbone is a 5 carbon sugar and is linked in beta linkage. And when we can have some decoration, some addition to structure, another 5 carbon sugar is arabinose, and it could be in the like when we number these carbons, this is 1 and this is 2. So arabinose could be in the 2 position or the 3 position, like in this figure. So, or it could be in both of them. So for arabinose islands, we know it's the xylose backbone. It can have some substitution, but then there is also potential for cross-linking. This is very important for the size of molecules. So cross-linking is through the esteric linkage here, connection, and then so we can have another arabinose island molecule that's parallel to here. And when we measure the size of the molecule, it's going to be this side additional to other side. So the molecule is going to be so big. As it gets very big, then it's going to be less and less soluble, and it's going to be even water insoluble. So if the arabinose island is water soluble or not soluble is going to be extremely important for the functionality of arabinose island, because they have different role. And most of the bakers, they are using enzyme cocktails to control the size of the arabinose islands. So in summary, now you know that arabinose islands are xylose and arabinose. They could be water soluble, water insoluble, and then the solubility is extremely important for the function. They have the cross-linking potential via the esteric linkages, ferulic acid linkages. If you look at arabinose islands, they are important for textural characteristics, shelf life. One unique feature of arabinose islands is the water holding capacity. They can hold water 10 times of their weight. For example, 1 grams of arabinose island can hold 10 times 10 grams of water. Is this good? It's good because higher the water absorption is better the baking quality indication for bread, and we can actually monitor that with farinograph water absorptions. But if there is a small size reduction, with the enzymes, or for some reason, if the molecule becomes smaller, that extra water could be released into dough system, and we can have sticky dough problems. So they will be very important for stability of the dough, again, how they interact with water. Cryostabilization for frozen dough. Again, for frozen dough, we really want to control the size of the ice crystals. How arabinose island is interacting with water is extremely critical for the stability of and the size of ice crystals in frozen dough. We will talk about them more, but the ice crystals, especially the large ones, are extremely detrimental for frozen dough quality. Why? Because they act as sharp knives and they chop and they cut the gluten network. So that's why we have to be very creative and careful in terms of stabilizing the ice crystals. Again, they have significant impact on the viscosity. And arabinose islands, they also have unique interactions with proteins, wheat proteins. They actually form three-dimensional gel systems. We are going to talk about them too. So arabinose islands, again, they can have different size and the high molecular weight versus the medium or low molecular weight. We can actually localize them and we can actually see them in the bread versus in the dough system. And here we have starch molecules are shown with green and the protein molecules are shown with red. And here we can actually see that the arabinose islands are interacting with proteins, and then the smaller medium-sized arabinose islands, they actually stabilize the gas cells. So one of the important studies we have done in the past, there are a series of studies, I'm only going to summarize very briefly, is remember that what arabinose islands are holding relatively higher amount of water. But again, if we have a smaller reduction in the size, then the water is being released. So this is a major problem in the refrigerated dough formulations. So refrigerated dough is different than frozen dough. The refrigerated dough is prepared very close to optimum water absorption, and the dough is stored at refrigerated temperatures. If there is a very small amount of enzyme activity that is hydrolyzing, reducing the size of arabinose islands, then we have water release into dough system. And actually that water is called dough syruping because there is a dark brown liquid formation, sometimes even visible by the consumers. So we want to control that water, and why we have that in the refrigerated dough formulations. So first we wanted to see how arabinose islands are impacting water absorption, water holding capacity of bread wheat. If you look at here, water absorption is 14% moisture basis in the control flour with no arabinose island, no additional arabinose island is 58, almost 58%, and the stability is 5 minutes. We are going to define what the peak time is, what the stability is in the following lectures. But here I want to put the emphasis on, as we increase the arabinose island content, there is increase in the water absorption. And then earlier in the arabinose island lecture, I talked about water soluble and insoluble arabinose islands. If you look at what are the functions of arabinose islands, this is a very nice cartoon summarizing the functionality of arabinose islands early in the fermentation and late in the fermentation process. Here the green bars are representation of water insoluble, or sometimes we call them water unextractable arabinose islands. And the blue is the water extractable arabinose islands. They are soluble. If we look at this figure, we can see that water extractable arabinose islands, which we know they are smaller than unextractable ones, they actually stabilize the gas cells. So they have positive impact on the loaf volumes and the quality of the bread. But the water unextractable ones, they actually destruct the gas cells. So they have detrimental effect on the gas cell formation, the stability of gas cells that are formed during fermentation process. This is one of the reasons the bakers are using enzymes. They have impact on reducing of water unextractable arabinose islands. So those enzymes, we call them xylanases. Sometimes they have different market name or commercial name, but the idea behind xylanases in the baking system is to reduce the amount of total water and extractable arabinose islands, reduce the size so we can have better gas cell stability in the bread formulations.